Okay, so the plan is today to hopefully get you through this meiosis and crossing over part of your notes, and then I'm going to come down hopefully after I get my kids going on some stuff, and we will spend some time looking at modeling crossing over, and then I'm hopefully going to be able to go over the very last page of this note guide looking at gene linkage and trying to figure all that. We'll figure that out together. That is the goal for today. Hopefully the weather cooperates and my kiddos down there with the regular bio kiddos cooperate too. So let's dig into meiosis. You may want to spend a little bit more time on your own. We're not going to spend a whole lot of time on meiosis, but we are going to look at these notes, and that's really about all we're going to do with it. But you do need to know some of the details of what's going on in here, so you know, especially as you're looking through your text review, make sure you're looking over that part in a little bit extra detail. So once again, we're looking at meiosis and crossing over. So you can kind of see in this picture here the different phases of meiosis. So we have similar order of phases where we go interphase, prophase, metaphase, anaphase, telophase. But the difference is this time we've got two divisions. All right, and so we've got really two main goals here with meiosis. We are trying to form haploid daughter cells. So when we're done, we'll have made 4 haploid daughter cells. So we have diploid cells. So di, what does di mean? Hopefully you said 2. Okay, so we have diploid cells. That means we have 2 of each kind of chromosome versus our haploid cells. They only have 1 of each kind. And we do meiosis to produce these haploid daughter cells because we need cells for our sex cells that are going to have half the normal number of chromosomes, because our sex cells, we think, okay, we have our egg and our sperm, and when they go to make the zygote, they go to make the baby, they combine. And we want to have normal babies that have 46 chromosomes. So in order to make that happen, we have to have our sex cells that have 23, or haploids. So we're going to look today, like I said, at how this process works. How do we go from being diploid cells to being haploid cells? All right, so here's a learning objective. This is really what we're looking at, specifically what we're trying to get you to be able to do for your AP test. So it says you need to be able to construct an explanation using visual representation or narratives. Okay, so be able to model this or explain what's going on here as to how DNA in chromosomes is transmitted to the next generation via mitosis or meiosis followed by fertilization. Okay, so today we're digging into meiosis. We've looked at mitosis. So speaking of mitosis, we're going to look at asexual reproduction in just a second. Another thing we want you to be able to do is connect meiosis with genetic diversity. All right, so to see what parts of meiosis provide new genetic varieties that are going to be used to help drive evolution. Okay, so we know in asexual reproduction, we don't have gametes. All right, we don't have those sex cells that are going to combine and form new offspring. So the genetic material only comes from one parent. And there are different forms of asexual reproduction. One of these forms is binary fission, which is done by bacteria, and they just copy their chromosome and then they split in 2. So you essentially end up with a clone. I'm going to try to make sure that you guys have enough time to write, since I know you guys are going to be watching this in class and not at home. So I'll try to slow down and pause. That way you can get these things written on the slide. If you need the sub to pause or go back, let him or her know, okay? All right, so asexual reproduction, essentially we're doing mitosis when we're doing asexual reproduction. So you start out with a diploid cell, you end up with a diploid cell. All right, and then we have sexual reproduction. So what is interesting, what we have to remember about sexual reproduction is that it doesn't mean that you have to have sex, all right? So most plants actually do sexual reproduction because the pollen is actually sperm, all right? So the pollen that gets drifted in the air, that blows in the wind, or gets on the bee's wings, is going to be sperm, and it's going to be deposited in the egg part of the plant. Okay, so it just means when we have two gametes that are going to fuse to form a zygote. Okay, so our gametes are going to be haploid. Sometimes we write this as 1N. So that N tells us the number of different types of chromosomes. So our gametes have one of each kind of chromosome. And then our zygotes, that fertilized egg there, is going to be 2N, or diploid, which means you have two of each kind, because you had one that came from the mom and one that came from the dad. So we know when we think about humans, we know we have 46 chromosomes and we have 23 pairs, right? That's where we get our diploid number from. We have 46 is our diploid number because we have 23 chromosomes that came from mom and 23 chromosomes that came from dad. All right, so how do we get the sperm and the egg? So this is a process called meiosis. And really the goal of meiosis is to reduce the chromosome number. So we have to divide that chromosome number in half when we go to form those gametes, to form the sperm and the ova, aka egg. Okay. Okay, and like we said before, really the reason behind this is we want to have normal babies, right? You guys are in the midst of your chromosomal abnormalities, like your karyotypes and all of that stuff, and you can see what big impact even just having one extra chromosome is, or having an extra piece is, and how those cause such problems genetically, especially for humans. Okay, a lot of times plants for some reason they don't really care as much. They can have whole extra sets of chromosomes, and that doesn't really bother them as much. But for humans, we're really sensitive having extra pieces or missing pieces of DNA. So really we want meiosis to go well. We want meiosis to correctly reduce the chromosome number to make sure our gametes are forming properly. Alright, so here's an overall picture of the first part of meiosis, what we've got going on here. So in interphase, we kind of skipped over interphase here, but in interphase, it's going to be doing what you would think of an interphase in the cell cycle, right? It's going to be having that G1 phase where it's going to grow and do its normal thing. And it's going to go through the S phase where it's going to copy the DNA, right? Same thing. We're going to make a copy of the DNA, prepare for division there. And then G2, the gap 2, where it's going to starting to really get ready to divide and copy those organelles. Okay, and then prophase, metaphase, anaphase, telophase. You can still think of those same words. The big thing here is you got to think about how the chromosomes are different. How do they look differently from mitosis? Okay, so prophase, same thing. They're still going to prepare to divide. We're still going to start having the chromatin. Hopefully you remember your chromatin. And those chromatin molecules are going to become chromosomes, right? So it talks about the homologous chromosomes are going to condense. Big thing here that is unique to this process you want to make sure you know this, is called crossing over. All right, crossing over happens in prophase 1. You're going to want to know when it happens. You're going to want to know what it means. So crossing over, well, I think we talk more about this later on, but crossing over is the idea we have these homologous chromosomes. The ones that are the same size, same shape, contain genes for the same traits. But one came from mom, one came from dad. We're going to switch pieces. We're going to switch parts. Okay, so a little piece of mom's is going to exchange with a little piece of dad's. Okay? And this is going to give you a new combination, something that has never been there before. Okay, so you're going to make a totally new combination there. So this is one way we're going to see genetic variety for meiosis. Okay, so that's happening in prophase. Same normal things that are happening in prophase. We're starting to see the spindle fibers form. We're starting to see the nuclear envelope disappear. Okay, and then we're moving into metaphase. Okay, so metaphase. What word do you think of when you think of metaphase? So we know middle, right? We're thinking about the middle. Okay, this time, instead of lining up in a single file line, they're going to line up side by side. Okay, instead of splitting into, when we go into anaphase, when we think about apart, instead of splitting the chromosomes apart into chromatids, we're just going to split the homologs or the homologous chromosomes apart. We're just going to separate those. So when you're done, you still have chromosomes instead of chromatid. All right, or instead of chromatids. Okay, so when we're done with the first phase, we have 2 chromosomes that are now genetically different because crossing over has happened. Okay, or we have 2 cells that each have 2 chromosomes. So we started out, I know it's kind of hard to tell in your prophase, but when you started, you had a diploid cell, you had your 2 in number was 4. Notice, when you're done with telophase, how many chromosomes do you have in each cell? 2, right? You have 2. So technically, this cell's haploid. So we could just be done. Right? We could just be done. But we're going to keep going because remember, we have these sister chromatids that we can separate even more. Okay, we can separate these chromosomes into chromatids and not really lose any genetic information. So we're going to do that in meiosis too. We're going to go ahead and even further separate them. So at the end of telophase 1, we're technically haploid, right? But we're going to go ahead and keep going. All right, so big thing here that also happens in anaphase 1. Like we said, the paired homologous chromosomes are going to separate and move toward the opposite poles. And at this point, they're not identical because of crossing over.